All right, well, let's go ahead and get started. Hi, everybody. I am Lauren Baker. I'm a K-12 scholar from UW-Madison, and welcome to our March K-12 seminar. I want to ask everyone to please take a moment and ensure that you are muted so that we don't have any disruptions during the presentation. We will have a question and answer session at the end of the talk, so please feel free to enter your questions into the chat box if you have them during the seminar, and that will help us kickstart the discussion at the end. We will have a post-event survey, so please do fill that out for us, as we appreciate and consider your feedback. So with that, I am honored to introduce our speaker for today, Dr. Michael Odom. Dr. Odom began his career as a paramedic. He spent a total of 8 years as a paramedic before and during his schooling, which I think is really cool. And he obtained his bachelor's degree in emergency medical care at Western Carolina University before he changed his focus to basic science research. Dr. Odom's PhD in physiology is from Brody School of Medicine at East Carolina University, where he studied neurological mechanisms of erectile dysfunction following treatment for prostate cancer. After his PhD, he moved on to Duke University, where he is currently a postdoctoral associate in the urinary dysfunction laboratory. He's currently studying diabetic bladder dysfunction, which is the most common complication of diabetes. He's going to talk to us today about how inflammation caused by an NLRP3-dependent mechanism can contribute to diabetic bladder dysfunction through effects on the urothelial barrier and smooth muscle function. So we look forward to hearing about all of that. So please take it away, Michael.
All right. Thank you for that introduction, and good afternoon, everyone. Again, my name is Michael. I'm just starting my third year as a postdoc and K-12 scholar here at Duke, working with Dr. Purvis and Dr. Hughes. And today I'd like to begin by thanking the organizers for allowing me the opportunity to present some of the findings from my K-12 project. So diabetes is one of the leading causes of mortality and morbidity in the nation today. The most common complication of diabetes is diabetic bladder dysfunction. In fact, over half of all diabetics will develop diabetic bladder dysfunction, and some don't even realize they're diabetic until these urinary symptoms occur. This is particularly troublesome since optimal glucose control does little to manage symptoms and there are no targeted therapies available. So in order to develop these much needed therapies, we must first understand how diabetic bladder dysfunction progresses. And we know inflammation is a key causative factor. One source of inflammation we're interested in is the NLRP3 inflammasome. This member of the innate immune system is ubiquitously expressed in bladder urothelial cells and activated by various damage-associated and pathogen-associated molecular patterns. Classical NLRP3 activation requires an initial priming step to upregulate transcription of pro-IL-1 beta and caspase-1. However, we have shown that urothelial cells do not require this initial priming step. Damage-associated and pathogen-associated molecular patterns directly oligomerize and form the active NLRP3 inflammasome. This activates caspase-1, which cleaves both pro-IL-1 beta and pro-IL-18. This results in a gasdermin D pore formation, pyroptosis, and a release of pro-inflammatory cytokines IL-1 beta and IL-18, which exacerbate bladder inflammation. Such NLRP3-mediated pyroptosis could be detrimental for bladder function. A breakdown in the urothelial barrier would permit noxious compounds in the urine to come in direct contact with underlying cells in the lamina propria and smooth muscle. And that brings us to the two aims we want to address. We aim to determine how NLRP3 activation during diabetes impacts urothelial barrier function and smooth muscle function. So we began by investigating the first aim. Like other epithelia, urothelial barrier function is regulated by key structural components of tight junctions, such as claudins and occludin. Meanwhile, components of adherence junctions, such as beta-catenin, regulate cell-to-cell adhesion. These components are responsible for forming barriers which prevent paracellular transport of various ions, urea, and small molecules. Now, bladder urothelia is also lined with protective apical uroplakins. These asymmetric tetramers form hexagonal plaques on the apical urothelial surface and provide an additional layer of defense. Now, we aim to specifically assess how diabetes-mediated NLRP3 activation impacts these key urothelial structure components and determine how barrier permeability is affected. Some evidence of urothelial damage and remodeling caused by diabetes has been shown in the literature. So previous studies using the streptozotocin-induced type 1 diabetic rat model show distinct structural damage to bladder urothelia. So after 3 weeks, the diabetic urothelia still closely resemble controls. However, by 9 weeks, dramatic changes are seen. Urothelia appear ragged and disorganized, and it becomes difficult to distinguish the boundaries of umbrella cells and intermediate cells. As indicated by the asterisk on the bottom row, areas of umbrella cell loss are evident, leaving the underlying cells exposed. We also see multiple regions of cell-to-cell junctions that are visibly broken. Now, this structural damage is only temporary. By 20 weeks, evidence of barrier repair and remodeling is noted. Umbrella cells and intermediate cells become more organized, and zippered membranes reappear at the umbrella cell periphery. Now, this clearly demonstrates that the urothelia undergoes extensive remodeling as diabetic bladder dysfunction progresses. And this was a fascinating study, which prompted a lot of further questions. For one, the mechanistic cause of this damage is largely unknown, as are the functional effects on barrier permeability and how permeability relates to specific urodynamic phenotypes. We hypothesized diabetes-mediated NLRP3-dependent mechanisms damaged urothelial structure, resulting in an increase in barrier permeability. So in order to address this hypothesis, we used a novel genetic mouse model. We crossbred type 1 diabetic Akita mice with NLRP3 knockout mice to yield 4 distinct groups of non-diabetic and diabetic mice with and without the NLRP3 gene. Now, at 15 weeks, female diabetic mice exhibit detrusor overactivity, defined by a decrease in void volume and an increase in voiding frequency. Interestingly, female diabetic mice lacking NLRP3 do not develop this overactivity phenotype. By 30 weeks, diabetic females have progressed to a detrusor underactivity phenotype, noted by an increase in void volume and a decrease in frequency. Again, diabetic females lacking the NLRP3 gene fail to develop this pathology. For our current experiments, we used the same 4 groups of female mice and evaluated barrier function at both the 15-week overactivity as well as the 30-week underactivity time points. We first wanted to measure urothelial barrier permeability. It was important for us to be able to measure permeability across the entire urothelia. To do this, we modified an existing ex vivo method and started by carefully separating the entire detrusor from the mucosal layer without damaging the urothelia. Once the detrusor was discarded, we tied a catheter to the remaining urothelial balloon just proximal to the ureters. Next, Evans blue dye was infused intravesically over the course of 10 minutes, and the urothelial preparation remained inflated for another 30 minutes. We then quantified how much Evans blue permeated through the urothelia and into the bathing solution. So first we see 15-week diabetic mice had nearly twice as much Evans blue permeate through the urothelia and into the bathing solution. Now this is indicative of barrier dysfunction. Specifically, this is an increase in small molecule permeability. Now, interestingly, diabetic mice without the NLRP3 gene did not exhibit this pathology. And we were excited to see that preventing a single pro-inflammatory pathway prevents this pathology from developing. And we then wanted to take another step further and determine what was the pattern of urothelial injury. Now, to determine this pattern of injury in vivo, we intravesically infused a solution containing the small molecule biotin. Similar to the ex vivo method, we allowed the small molecule containing solution to instill for 30 minutes. Bladders were then excised and processed for histology. Now, slides were labeled with fluorescent streptavidin to detect this biotin, and zonoocclutin-1 to identify the urothelial layer. We then quantified the percentage of urothelium which biotin permeated through. Here we see white arrows indicate regions of barrier damage in which the intravesically administered biotin was able to penetrate through the urothelial layers and accumulate in the submucosa. This magnified inset at the bottom shows this region in better detail. Now, at 15 weeks, we see few regions of biotin penetration in our non-diabetic animals. And this is in stark contrast to our diabetic animals. In 15-week diabetics, we see numerous patchy regions of barrier damage indicated by white arrows. The insets below show 2 magnified images of barrier damage and this urothelia containing biotin in the submucosa. Urothelial layer thinning is noted in some of the damaged areas. And interestingly, if you look at the region indicated by the asterisk, we also see evidence of apical urothelial cell loss. We then looked at the bladders of non-diabetics and diabetics without the NLRP3 genes. In both samples, few areas of barrier damage are noted, and no immediately obvious differences were noted between these two groups. Now, when the data is analyzed, it's clear that diabetes causes significant barrier damage at the 15-week overactive time point. Patchy areas of barrier damage account for over half of the total urothelia. Interestingly, in diabetics lacking the NLRP3 gene, we don't see this pathology. Preventing NLRP3-mediated inflammation by deleting the NLRP3 gene preserves barrier function in diabetic mice at the overactive detrusor time point. And we are excited again to see that preventing a single pro-inflammatory pathway prevents this pathology from developing. And we then wanted to take another step further and determine how urothelial structure components were affected. In separate experiments, we isolated the mucosal layer and measured barrier gene expression using qPCR. Specifically, components of uroplakins, tight junctions, and adherent junctions were measured. At this time point, diabetes significantly downregulates the expression of uroplakin components UP1B and UP2. However, ablating the NLRP3 gene preserves uroplakin gene expression in diabetic bladders. A similar pattern occurs with the zona-occludin gene expression. Expression of these tight junction components are significantly downregulated as a result of diabetes. Zona-occludin expression is preserved, though, in diabetics lacking the NLRP3 gene. Likewise, diabetes significantly downregulated barrier-forming claudin 4 and claudin 8 gene expression. And again, diabetics without the NLRP3 gene don't demonstrate this pathology. At this 15-week overactive detrusor time point, no changes in gene expression of the adherence junction component beta-catenin were noted. So overall, at this 15-week detrusor overactivity time point, diabetic mice show an increase in urothelial permeability and a decrease in barrier genes. NLRP3 gene ablation prevents this pathology. But what about the underactive time point? We conducted the same experiments in 30-week female mice that exhibit an underactive detrusor phenotype. Again, we turned to ex vivo methods to measure permeability across the entire urothelium. Surprisingly, barrier permeability in our 30-week diabetic group was no different than controls, and NLRP3 deletion had no further impact on barrier permeability. Now, initially, we found this very odd. It would be logical to presume pathology worsens as a result of a chronic condition such as diabetes, but here that isn't the case. When we performed our assessment of in vivo barrier function, we found a similar pattern. In all four groups of non-diabetics and diabetics with or without the NLRP3 gene, we see very few regions of urothelial barrier damage. Ultimately, we found no evidence of barrier damage in our 30-week groups. Even though there were no functional changes in barrier permeability, we then wanted to determine if barrier gene expression was affected. Now, even though uroplakin gene expression was downregulated in 15-week diabetics, no downregulation is observed at 30 weeks, regardless of the absence or presence of NLRP3. At this time point, there were also no differences in zona-occludin expression either. Now, there were no significant differences in the expression of barrier-forming claudins. And again, this was very surprising, and we began questioning how the urothelia was able to overcome such an initial insult at 15 weeks and undergo either extensive repair or remodeling by 30 weeks, despite the constant presence of diabetes. One clue may be the upregulation of beta-catenin at 30 weeks. Now, this component of adherence junctions is responsible for maintaining cell-to-cell adhesions, but also plays a key role in urothelial cell proliferation, particularly through the beta-catenin WNT signaling pathway. In diabetic mice lacking NLRP3, this upregulation does not occur, which makes sense considering this group did not show any evidence of barrier damage. So by 30 weeks, diabetic mice with detrusor underactivity no longer demonstrate changes in barrier permeability or downregulation of barrier gene expression. This suggests urothelial repair or remodeling has occurred between the overactive and underactive time points. In our animal models, we conclude diabetes causes NLRP3-dependent barrier dysfunction associated with detrusor overactivity, but not underactivity. And so we submitted a manuscript of our barrier function findings and moved our attentions towards the second aim. Now that we know the urothelial barrier is compromised at an early time point, we really wanted to know how diabetes and NLRP3 activation impact bladder smooth muscle function. And we began by asking a simple question: what NLRP3 dependent signaling pathways are dysregulated by diabetes? Now, currently we have 2 ongoing projects evaluating smooth muscle function at the overactive 15-week time point, as well as the underactive 30-week time point. And we are specifically measuring smooth muscle contractility ex vivo using traditional organ bath studies. Essentially, we cut strips of longitudinal bladder smooth muscle, and once cut, these strips are connected to a force transducer on one end and an adjustable clamp on the other end, while being maintained at 37 degrees in an aerated physiological buffer. After the tissue has been acclimated and baseline tension has been added, we either add drug compounds or electric current into the buffer solution and measure changes in contractile force. We use this approach to determine how various signaling pathways become dysregulated and contribute to diabetic bladder dysfunction. Now, the main signaling pathways responsible for bladder contraction are the cholinergic and perinergic signaling pathways. Cholinergic signaling refers to the release of acetylcholine and the effect of acetylcholine on both muscarinic receptors, and perinergic signaling involves the release of ATP and the effects on perinergic receptors. Both compounds are released by afferent autonomic nerve terminals, and both compounds are also released by the urothelia in response to various stimuli such as stretch. Interestingly, both compounds are not released in equal amounts. The dependency of cholinergic versus perinergic contribution to bladder contractions varies depending on sex. In males, it's commonly accepted that over 90% of bladder contractions are mediated by cholinergic pathways. On the other hand, in females, cholinergic signaling contributes much less, and perinergic mediated contractions have been shown to be responsible for up to 50% of bladder contractions. Now, in these studies, we are able to cause a release of neurotransmitter from the nerve terminals, which are still attached to the bladder tissue, by using electric field stimulation and delivering electric charge. One quick technical note is that all of the EFS contractility data I'm showing today is tetrodotoxin sensitive, which simply means that the contractions are in fact due to the release of neurotransmitters. And we performed these experiments in the presence and absence of the muscarinic receptor antagonist atropine to determine the cholinergic contribution, as well as the perinergic P2X1 receptor desensitizer, alpha beta methylene ATP, to determine perinergic contributions. We began with our 15-week group in which diabetic mice demonstrate an overactive phenotype. Using denuded bladder strips, we maintained a constant voltage and duration and applied stepwise increases in frequency to cause neurotransmitter release and smooth muscle contraction. Now, as we increase these frequencies, the neurotransmitter-mediated contractions of our non-diabetic group with functional NLRP3, shown here as the white boxes, appear to plateau. As for the diabetic detrusors, shown as black boxes, we see diabetes significantly increases neurotransmitter-mediated contractions at the higher frequencies. Now, this is to be expected since diabetic mice at this time point have an overactive detrusor phenotype. We then incubated the same tissue strips and atropine and applied electric field stimulation again to determine if the increase in contractility is due to cholinergic signaling pathways. When we inhibited muscarinic receptors with atropine, we no longer see this diabetes-mediated increase in contractility. This tells us that diabetes increases bladder contractility through dysregulation of cholinergic signaling pathways. Now, to further identify the perinergic contribution, we then blocked muscarinic receptors and desensitized the perinergic P2X1 receptor, which is thought to be the primary perinergic receptor responsible for bladder contractility, and then repeated electric field stimulation in the same strips. Though perinergic signaling contributes greatly to the contractile response, no differences are noted between non-diabetic and diabetic smooth muscle. And this indicates that cholinergic contributions, not perinergic, are responsible for the increase in smooth muscle contractility. We then repeated these experiments in non-diabetics and diabetics lacking the NLRP3 gene. Interestingly, NLRP3 gene ablation did not seem to affect this pattern at first. Denuded diabetic detrusors still develop an increase in cholinergic activity and increase in contractility. And this was initially disheartening, but it's important to reiterate that NLRP3 is ubiquitously expressed within the urothelia. Equally important is the fact that bladder smooth muscle contractility is dependent upon vasoactive molecules released from the urothelia. And since we are studying how inflammation in the urothelia impacts bladder function, we simultaneously performed these experiments in bladder strips with an intact mucosa. Now, with an intact mucosa, diabetes still increased contractility mediated by cholinergic neurotransmitters, but did not impact perinergic signaling. And this was the same pattern observed in our denuded detrusors. However, without functional NLRP3, diabetic bladders with an intact mucosa demonstrate no changes in neurotransmitter-mediated contractions. Even though the denuded detrusors of diabetic NLRP3 null mice show increased contractility, the mucosa mitigates such damage. And ultimately, we see no differences between the non-diabetics and diabetics without NLRP3. We again blocked muscarinic receptors with atropine and found that without NLRP3, there were no differences in the contribution of cholinergic and signaling in diabetics with an intact mucosa. We then blocked the muscarinic and perineurgic receptors with atropine and alpha-beta methylene ATP and repeated electric field stimulation. Now, this is really exciting for us. The statistically significant decrease in diabetic NLRP3 null contractility indicates the perineurgic contribution is much higher than in non-diabetic NLRP3 null mice. As for the cholinergic, our EFS data showed a diabetes-mediated increase in contractility due to increased cholinergic signaling activity. From that data alone, we can't tell if this is due to an increase in acetylcholine release, modification of acetylcholine after being released, or an increase in muscarinic receptor or sensitivity. To determine specifically how muscarinic receptor-mediated contractions are impacted, we constructed concentration response curves to a muscarinic receptor agonist, carbachol. When we look at bladder strips from non-diabetic and diabetics with NLRP3, we see that diabetes does not impact muscarinic receptor-mediated contractions at this 15-week time point. And this holds true regardless of whether the mucosa is intact or not. In diabetics lacking NLRP3, denuded detrusors show an increase in muscarinic receptor-mediated contractions. However, when the mucosal layer remained intact, no observable differences in muscarinic receptor contractions were observed, signifying that urothelium mitigates this pathology. And finally, we measured contractility mediated by alpha-beta methylene. So this drug is unique in that it activates the perinergic P2X1 receptor to cause bladder contractions before the compound then causes receptor desensitization. So essentially, this drug works as both an agonist and inhibitor. Now, here we see that diabetes does not impact P2X1 receptor-mediated contractions at this time point. I believe the NLRP3 gene did not further impact P2X1 receptor contractility. And this is particularly interesting. Our EFS data showed that in the absence of NLRP3, neurotransmitter-mediated contractions of diabetic bladders have an increased procontractile contribution from perinergic signaling. Here, we stimulated the main P2X1 receptor and found no differences, suggesting that either the receptor sensitivity or receptor population are not affected by diabetes. This increase in perinergic signaling among diabetics lacking NLRP3 is likely due to either an increase in ATP release or modification of ATP after being released by ectonuclease activity or other mechanisms. And this is an ongoing project, and so far at this overactive 15-week time point, we can make a couple of conclusions. Diabetes increases cholinergic-mediated contractility, likely due to an increase in acetylcholine released from nerve terminals. NLRP3 contributes to this pathology, and in the absence of NLRP3, diabetic urothelia mitigate this damage by either reducing the amount of ATP released or via ATP modification after the release. So in addition to the 15-week data, we are simultaneously evaluating how diabetes and NLRP3 impact smooth muscle function at the 30-week underactive detrusor time point. And we repeated the exact same set of experiments in our 30-week mice to evaluate how diabetes impacts cholinergic and perinergic signaling. And frankly, the results were quite surprising. We did not find any ex vivo changes in cholinergic or perinergic contractility in diabetic mice with an underactive detrusor. I won't go through all the negative data, but here we see moving from left to right that there are no changes in neurotransmitter-mediated contractions. There were no changes in muscarinic receptor-mediated contractility to carbachol, and there were no changes in perinergic P2X1 receptor-mediated contractions. We performed additional studies aimed towards elucidating the effect of other perinergic receptors and the role of ectonucleases, but ultimately we came to the same conclusion. We simply did not find any ex vivo cholinergic or perinergic dysfunction. So this led us to explore other signaling pathways, most notably the prostaglandins. There's been quite a bit of literature regarding how diabetes impacts prostaglandin release. Some studies have shown that prostaglandin concentrations are higher in urine from diabetics with an overactive bladder, but lower in diabetics with an underactive bladder. But prostaglandins are known potent vasoconstrictors released from both the detrusor and urothelium. in various disease states, inflammation alters the production and release of prostaglandins. And much of this work involving the bladder has focused on prostaglandins E2 and I2. Now, both PGE2 and PGI2 activate the EP family receptors on smooth muscle to facilitate these contractions. Interestingly, recent studies have shown that prostaglandin F2A also has potent procontractile effects on bladder smooth muscle, and this is via FP receptor activation. Now, the role of bladder FP receptor activation during diabetes has yet to be defined. So we first want to determine if there are any functional differences in smooth muscle contractility or prostaglandins. To do this, we turn back to our ex vivo tissue bath experiments. We specifically focused on PGE2, which activates the EP receptors, and PGF2A, which predominantly activates the FP receptors. Now, to determine if the effects of PGF2A were due solely to FP receptor activation, we repeated the PGF2A curve in the presence of a partial FP receptor antagonist, AL8810. Now, we started with our EP receptor agonist, prostaglandin E2, and found that contractions mediated by PGE2 were not affected by diabetes and bladder shifts both with and without the mucosa. In the absence of NLRP3, diabetic detrusors showed increased in PGE2 contractility. Interestingly, though, when the mucosa was left intact, these changes were no longer present. Again, urothelia mitigated the changes in detrusor contractility. So next, we determined how diabetes impacts bladder contractions mediated by the FP receptor agonist, prostaglandin F2A. Now, much to our surprise, the underactive diabetic detrusors actually demonstrate increased contractility to PGF2A. Now, with an intact mucosa, these effects are further amplified, and we'll talk more about the relevance of this in a bit. But NLRP3 null diabetics at this time point failed to develop a pathologic underactive detrusor phenotype. And here we see that they also do not develop this increased contractile response to PGF2A. So that brings us back to this graph. The fact that an underactive bladder is hypercontractile to an FP receptor agonist suggests that such an agonist may have therapeutic potential to treat existing underactive bladder symptoms in diabetic patients. So now our mindset has shifted from determining which signaling pathway is dysregulated by diabetes to more of a mindset where we need to determine if an FP receptor agonist would be an effective treatment for underactive bladder symptoms. Now, the first step is to determine if these contractility differences are mediated specifically by FP receptor activation. And to do this, we incubated the same strips in a partial FP receptor antagonist, AL8810. FP receptor inhibition abolished the increased contractile response to PGF2A in diabetic bladders. And this is very exciting for us because now we are cautiously optimistic that we have a pharmacological target to treat existing underactive bladder symptoms in diabetic patients. And that's really the big takeaway so far with our 30-week data. At this underactive phase, there were no changes in cholinergic or perineurgic contractility, but diabetic detrusors have increased FP receptor contractility, suggesting the FP receptor may be a therapeutic target. And I'll end this talk today with an intriguing case report. Before we began administering a FP receptor agonist to our diabetic mice to treat this pathology, we searched the literature for any evidence that this is even feasible or effective. And that's when we came across this case report. There are several FP receptor agonists on the market today. Most, such as latanoprost, are available as eye drops used to treat glaucoma. Now, in this case report, a 62-year-old female began taking latanoprost and developed urge incontinence. And one of the most interesting quotes from this report was, her symptoms included a strong urge to pass frequent small amounts of urine and inadvertent leak of urine. When she stopped taking latanoprost, the overactive bladder symptoms subsided, but symptoms returned when she resumed latanoprost treatment. So even though this is an N of 1, it does provide proof of concept that in vivo administration of an FP receptor agonist can augment human bladder function. So thank you all for your time, and I'll be happy to answer any questions.